Assalamu alaikum. In this lecture, we will continue to talk about the gram-positive bacilli. As we know, the bacilli are classified into gram-positive and gram-negative bacilli. The gram-positive bacilli are further classified into non-spore-forming, as we mentioned the Corynebacterium and Listeria, and the spore-forming gram-positive bacilli. The spore-forming gram-positive bacilli are further classified into aerobic, which means they need oxygen for their growth, and anaerobic gram-positive bacilli. The aerobic gram-positive bacilli includes the genus Bacillus, while the anaerobic includes the genus Clostridium. So, in this lecture, we will talk about the family Bacilli genus Bacillus, which are aerobic and facultative anaerobic spore-forming gram-positive bacilli. The genus Bacillus comprises more than 50 species of the gram-positive bacilli spore-forming. They are large. They are the largest gram-positive bacilli, among the largest gram-positive bacilli. Catalase positive gram-positive rods that form endospores, and the rods are arranged in chains. The endospores are present inside the bacillus and not bulging the mother cell. They are not stained by gram stain. So the spore appear as colorless area inside the bacillus, and usually the bacilli are arranged in chains, which give it the bamboo stick appearance. تشبه عود البامبو. The genus Bacillus, on gram staining, the spores don't stain and appear as empty space and doesn't distend the vegetative cell, means that the spore inside the bacillus not distending the bacillus within the limits of the bacterial cell wall, while in Clostridium the spore is bulging, distending the mother cell. The spore is bulging in genus Bacillus, in genus Clostridium, while in genus Bacillus the spore is non-bulging, not distending the mother cell. The spores are stained using special spore stain, which is called the spore stain, and they are resistant to environmental change. The spores are a very resistant structures, resist many chemicals and heat to certain temperatures and disinfectants for certain periods. They persist for years in the dry earth, and they are the commonest contaminant in bacteriological culture media. When we prepare culture media, the most important contaminant present in the culture media is genus Bacillus. The spores are present in soil, dust, water, and air everywhere, and they are the major contaminant of bacteriological culture media. The spores are heat resistant, so it can resist the severe environmental conditions like nutritional deprivation, temperature extremes, drying, and so on. So when the culture medium devoid of nutrient substances, it will form the spore. This is the mechanism of formation of spores from vegetative cells. First we have, this is the vegetative cell, and the reproduction of this vegetative cell by binary fission, and when there is nutrient substances, it will continue the cycle with the vegetative cycle, while when there is abnormal temperature, decrease in nutrient substances, or dryness, it will change to the spore formation, and this is the mechanism of spore formation. And finally, the vegetative mother cell will disappear, and only the spore will stay in the environment, and it could stay for years on earth, on soil, dust, water, and air. When there is favorable conditions for the growth, this spore will change to the vegetative cell. The species which are most important isolated from human infections, first, the most important pathogenic species is Bacillus anthracis, which is the most important pathogenic species, which causes anthrax. بنسميها الجمرة الخبيثة. And it's the principal pathogen of the genus. While Bacillus cereus, it's less pathogenic, and it may cause food poisoning. It may produce severe infection in immunocompromised humans. Any immunocompromised human are prone to infection with even normal flora. So they are causing food poisoning, and immunocompromised may cause severe infections. While Bacillus subtilis is a saprophytic organism isolated from the environment from contaminated culture media and prevalent in soil, water, and air. It's not pathogenic. It's present as a normal saprophytic microorganism. This is the microscopical appearance of genus Bacillus in the vegetative form, and the spores are present inside the bacillus, and this is the chain of the bacilli, which give the microorganism bamboo stick appearance. They are gram-positive, spore-forming. The non-pathogenic species are motile, while the pathogenic species non-motile. Only Bacillus anthracis is not motile, while the other species are motile. This slide is showing the gram stain microscopical appearance of the genus Bacillus in gram stain. We will see that the bacilli are gram-positive, and there is an empty space, unstained space inside the bacillus, which forms the site of the spore. They are arranged in chains, and the microorganism is rectangular in shape. While the other smear is stained by malachite green stain. Here we will say that the microorganism is stained red and the spore green inside the bacillus. We shouldn't mention that the microorganism is gram-positive or gram-negative in this slide, because this is a malachite green spore staining, not a gram stain. Bacillus anthracis was the first pathogenic bacteria observed under the microscope. Bacillus anthracis is one of the largest pathogenic bacteria, gram-positive, non-acid-fast, non-motile. The non-pathogenic species are motile, while the pathogenic Bacillus anthracis is non-motile, citrate, and spore-forming. The bacillus is rectangular in shape, as we mentioned. It's rectangular in shape and arranged in a filamentous chains in culture. The rectangular shape and arrangement in chains with end-to-end long chains give the microorganism bamboo stick appearance. The spore in Bacillus anthracis is oval, central in position, while in the non-pathogenic species it's not central in position. And in both cases, the spore not swelling the mother cell, while in genus Clostridium, the spore is bulging, swelling the mother cell. The spores are formed in culture, in the soil, but never in blood or tissue of living animals. When we isolate the microorganism from the tissue, in tissue it not form spores, only in the environment or in culture. This is the bamboo stick appearance on gram staining of Bacillus anthracis, and its shape is similar to the bamboo stick. The microorganism is aerobic or facultative anaerobic, which means it can grow in the presence and absence of oxygen in a temperature range from 15 to 45 degrees centigrade. On nutrient agar, the colonies are round or irregular in shape. Its edges are irregular. The edges of the colony are irregular, not completely round. And the colony is elevated slightly, 2 to 3 millimeter in diameter in size, raised, dull, opaque, and grayish to white in color, with a frosted glass appearance. The shape of the colony is similar to a frosted glass, and the edge of the colony resembles locks of matted hair. The edge of the colony similar to locks of matted hair, which give the description of the colony as medusa head appearance, which is seen under the low power microscope. When examine the edge of the colony, we will see this medusa head appearance, like matted hair appearance, or named as medusa head appearance, which is seen under the low power microscope. So the colonies are irregularly round, 2 to 3 millimeter in diameter, as we see that the colonies are larger than those seen in Streptococcus and Staphylococcus, raised, dull, opaque, grayish white, and having a frosted glass appearance. And the edge of the colony, when examined under the low power of the microscope, we will see this characteristic medusa head appearance. Under oil immersion, when we examine a smear prepared from these colonies, we will see the characteristic non-positive bacilli, spore forming, and arrangement of the microorganism in chains. So this matted hair under low power is called the medusa head appearance. This is the frosted glass appearance. The shape of the colony is similar to frosted glass. This growth is on a blood agar. On a blood agar, the colonies are non-hemolytic, while the non-pathogenic species are hemolytic. So the differences between the pathogenic genus Bacillus anthracis, and the non-pathogenic species. First, the pathogenic species, non-motile, non-hemolytic. The non-pathogenic species, like Bacillus subtilis, cereus, and the other strains, they are hemolytic and motile. The pathogenic species, non-hemolytic and non-motile. The third culture medium used in a tube is gelatin medium. In a gelatin stab, there is growth down the stab line with lateral spikes, which give the growth a characteristic inverted fir tree appearance. This is the characteristic inverted fir tree appearance, which is characteristic for the growth of Bacillus. The selective culture medium used is a PLET medium, which is the abbreviation of polymyxine lysozyme EDTA thallose acetate. PLET medium has been advised to isolate Bacillus anthracis from mixtures containing other spore-bearing bacilli, so is a selective culture medium for Bacillus anthracis. It contains polymyxine lysozyme, ethylene dioxide, tetraacetic acid, and also EDTA. So it will inhibit the growth of other bacteria and only favors the growth of Bacillus anthracis. Bacillus anthracis have a characteristic ability to form spores. These spores are resistant to many environmental conditions. and temperature range. With moist heat, the vegetative bacilli are killed. The original bacilli are killed at 60 degrees centigrade in 30 minutes, while the spores in moist heat remained and only killed at 100 degrees centigrade for 10 minutes. At temperature 100 degree for 10 minutes, after that it will be killed. While using dry heat, the spores are killed at 150 degree in 60 minutes. At temperature 150 degree in 60 minutes. The spores are also killed by 4% formaldehyde or 4% potassium permanganate in a few minutes. The microorganisms sensitive to benzyl penicillin, streptomycin, tetracycline, chloramphenicol, ciprofloxacin, cephalosporins, and sulfonamides. So these are used in the treatment of the infected patients. As long as genus Bacillus anthracis is a virulent strain, it means that the microorganisms have a virulence factors. The most important virulence factors is first the capsule, which interferes with phagocytosis. So the most important function of the capsule in any microorganism is that interference of phagocytosis. While the strains that not produce the capsule are not virulent because they are phagocytosed and the body can attack them and they are eliminated soon. The most important pathogenic virulence factor is the anthrax toxin, which consists of 3 proteins: lethal factor, protective antigen, and the edema factor. These 3 are parts of the anthrax toxin: the lethal factor, protective antigen, and edema factor. Each one alone is not effective, not toxic, each of which individually is non-toxic, but together they act synergistically to produce damaging effect. Means that the lethal factor with the protective antigen, they both will form the lethal toxin, which have a lethal toxic effect on the tissues and cells, cause tissue damage and shock. The protective antigen with the edema factor, both will form the edema toxin, which will cause the edema which occurs in case of anthrax. So each one alone is not toxic. Lethal factor only is not toxic. The protective antigen is not toxic alone. The edema factor alone is not toxic. But the combination of lethal factor with the protective antigen or a protective antigen with the edema factor will cause these powerful toxins, the edema toxin and the lethal toxin. The edema factor with the protective antigen will form a toxin known as the edema toxin, responsible for cell and tissue edema, while the lethal toxin is a major virulence factor, cause death in infected animals and humans. So these are the important virulence factors in the Bacillus anthracis. This is the edema factor and the lethal factor when entering the cell and exerting their effect by increasing cyclic AMP, causing edema, or the lethal factor exerting its effect, causing necrosis and hypoxia. Pathogenesis and clinical infections caused by anthrax. Anthrax is a zoonosis, which means originally it's a disease of animals, and the humans are infected accidentally or in an occupational form. Cattle, sheep, goats, and other herbivores are naturally infected. The disease not spread from animal to animal, only through ingestion by these animals of food containing the spore. After ingestion, they will get the infection. Anthrax is endemic in developing countries, and the human become infected incidentally by contact with infected animals or their products, or sometimes by insect bites. There are 4 types of anthrax infecting human, either cutaneous, ingestion, inhalation, or injectional anthrax. Cutaneous, inhalation, ingestion, or injectional anthrax. Inhalation, by inhalation of spores, particularly in patients with wool sorting, so it's called wool sorter's disease, and the spores enter inside the body and exert its effect in the lung. The cutaneous by skin abrasion in peoples dealing with infected animal products, and usually it occurs in exposed areas of the body, in the hands, arms, or the face. The ingestional gastrointestinal anthrax, by ingestion of contaminated food with these spores. In cutaneous anthrax, it begins 2 to 5 days after infection. Usually the lesion begins as a small papule. This papule develops within a few days into a vesicle containing dark fluid, bluish-black fluid, which ruptures, and the vesicles will form this black scar at the base of the lesion, surrounded by small vesicles, which is very prominent inflammatory ring around the lesion. This black scar is a characteristic for a lesion caused by Bacillus anthracis. It's a characteristic for cutaneous anthrax. It's sometimes referred to as malignant pustule because of black color. The lesion classically found in the hand, forearm, or head and is painless. It occurs in the exposed area of the body when dealing with animal products. Cutaneous anthrax generally resolves spontaneously, but 10 to 20% if untreated patients may develop to fatal septicemia and meningitis due to spread of the microorganism inside the body. These are other pictures of cutaneous anthrax. We will see this characteristic black lesion surrounded by edema and vesicles. This is the characteristic black scar or malignant pustule. The second disease caused by Bacillus anthracis is pulmonary anthrax. Pulmonary anthrax, also known as wool sorter's disease. It used to be common in workers in wool factories. Due to inhalation of dust from infected wools, this is a hemorrhagic pneumonia. When there is inhalation of these spores, it will cause hemorrhagic lesions inside the lungs and causing hemorrhagic pneumonia with a high fatality rate. It may also cause hemorrhagic meningitis, which occurs as a complication. The spores from the dust of wool are inhaled, phagocytosed in the lung, and transported by the lymphatic drainage to the mediastinal lymph nodes. And these are will cause germination, forming vegetative bacilli, followed by toxin production and development of hemorrhagic mediastinitis and sepsis, which are usually rapidly fatal. Important thing in this route of infection is that it may be used as a bioterrorism agent. In the US, a bioterrorism inhalation cases in 2001, and the pathogenesis was the same as in inhalation anthrax from animal products. It resulted in 22 cases, and 11 are inhalation and 11 are cutaneous. It occurs by receiving males and the papers of the male are contaminated with the powder containing spores of this microorganism. The third disease entity is intestinal anthrax. Intestinal anthrax is rare and occur mainly in primitive communities who eat the carcasses of animals dying of anthrax. An individual may suffer after a day or so from hemorrhagic diarrhea and dies rapidly from septicemia. There is either oropharyngeal lesion or abdominal lesion, which will cause severe infection and toxemia and may cause septicemia and death. The fourth form of the disease is the injection anthrax. It has caused outbreaks among persons who inject heroin that has been contaminated with spores. It's characterized by extensive, painless subcutaneous edema and the notable absence of the scar characteristic of cutaneous anthrax. There is no blackish scar. The lesion characterized by diffuse edema and is painless, extensive subcutaneous edema. There is no scar in this lesion because the spore enters deep with the injection. Patient might progress to hemodynamic instability due to septicemia. So how we diagnose a patient infected with Bacillus anthracis? First, the specimen. In laboratory diagnosis, first the specimen. The specimen should be material from the infected area. In cutaneous anthrax, then the malignant pustule should be swabbed from underneath the lesion, while in pulmonary anthrax, sputum is the preferred specimen. In gastrointestinal anthrax, either gastric aspirate, feces, or food, in this case, will be the appropriate specimen. And the blood in septicemic stage of all forms of infection is a preferred specimen. Specimen should be taken before antibiotic therapy has been instituted. Microscopical examination of the specimens collected. We prepare smears of each specimen and stain with gram stain, which show typical large gram-positive bacilli. Direct fluorescent antibody test for capsule and for polysaccharide cell wall antigen confirm the identification. Culture used in the cultivation of the microorganism, nutrient agar, blood agar, and the PLET media. On nutrient agar, they grow and give us the characteristic large, irregular in shape colonies with a frosted glass appearance, and the edges of the colony are irregular with medusa head appearance. On a blood agar, the same growth, but they are non-hemolytic. On a blood agar, the pathogenic species is non-hemolytic. And on a PLET medium, when it grow, it means that the microorganism is Bacillus anthracis because it's a selective culture medium for Bacillus anthracis. We prepare smear from the culture and look for chains of long, large gram-positive bacilli, and the spores is central, oval, non-bulging spore. This is spore staining showing the microorganism inside the bacillus, the spore inside the bacillus, which is stained green in color. The spore is green in color, and the bacillus is stained with saffronin red in color. This is not a gram stain. This is spore staining method. Confirmatory tests, we use biochemical and physiological reactions, demonstration of non-motility. Bacillus anthracis is a non-motile microorganism. Gelatin liquefaction positive in case of Bacillus anthracis, and on gelatin stab there is a characteristic inverted ferretry appearance, and this will identify Bacillus anthracis completely. Toxin production can be demonstrated by immunological or gene probe methods in reference laboratories. Fifth, the serological diagnosis. by ELISA is seldom used diagnostically. And lastly, PCR is a sensitive and specific technique uh, have been developed for the detection of anthrax contamination of animal and agricultural products. This slide is stained with spore staining method. We will see the spore is stained green in color and the bacillus rectangular arranged in chains, and they are red in color. This is the characteristic growth on blood agar. Give us a characteristic glass, frosted glass appearance, and medusa head colonies on blood agar. They are non-hemolytic. And this is gelatin stab with inverted fir tree appearance. Treatment is by ciprofloxacin, which is the drug of choice. Penicillin, doxycycline, erythromycin, or chloramphenicol can be used. Chloramphenicol used in susceptible people to penicillins. Immunization using live attenuated bacilli. They are anthrax bacilli attenuated by growth at high temperature. So this is vaccine is from the bacillus itself, while the alum precipitated toxoid is prepared from the toxin, from the protective antigen protein of the toxin, which have been shown to be a safe and effective vaccine for human use. The first vaccine is used in animals, while the toxoid vaccine is used in humans. It has been used in persons occupationally exposed to anthrax infection. Prevention and control of infection, of course, by disposal of animal carcasses or by burning or by deep burial in lime pits. The decontamination of animal products, protective clothing and gloves, active immunization of domestic animals, and persons with high occupational risk should be immunized. The other species in this genus, in genus Bacillus, is Bacillus cereus. Bacillus cereus produces toxins that cause disease that is more of intoxication than a foodborne infection. It's not an infection, it's an intoxication. It mainly causes food poisoning, which is caused by this microorganism, and it has two distinct forms. The emetic type, which is associated with fried rice, milk, and pasta. It's manifested by nausea, vomiting, abdominal cramps, and occasionally diarrhea. And usually it's a self-limiting, and the recovery occurs within 24 hours. It begins 1 to 5 hours after ingestion of the food containing the emetic toxin in contaminated food products. When rice are cooked and allowed to cool slowly, the spores will germinate and the vegetative cells will produce the toxin during the log phase growth or during the sporulation. So the main symptoms in this type of infection is vomiting, abdominal cramps, and occasionally diarrhea. The other form of intoxication caused by this microorganism is the diarrheal type, which is associated with meat dishes and sauces. It has an incubation period of 1 to 24 hours and manifested by profuse diarrhea, abdominal pain and cramps, and fever. The fever and vomiting are uncommon, but they may occur. The other disease caused by this type of infection of microorganism is the eye infection. Here, the emetic form is caused by consumption of infected rice with the bacillus, and it will form the toxin inside the rice, while the diarrheal form is caused by meat and its products. The eye infection also may occur with the severe keratitis. Also, the microorganism may cause localized infection as wound infection, endocarditis, catheter-associated bacteremia, and other instrumentation-associated infections. Bacillus cereus is resistant to a variety of antimicrobial agents including penicillins and cephalosporins على عكس الباسيلس anthracis اللي تكون sensitive to most of these antibiotics. So serious non-food-borne infections should be treated with vancomycin or clindamycin with or without aminoglycosides. Ciprofloxacin has been useful for the treatment of wound infections caused by this microorganism. On blood agar, the microorganism is beta-hemolytic, while Bacillus anthracis is a non-hemolytic non-motile microorganism. Bacillus cereus is motile and cause beta hemolysis on blood agar. These questions should be answered by you after the lecture, والإجابات ممكن تنشروها على داخل المنشور بالتعليقات. يعني المفروض اللي يفتهم المحاضرة يقدر يجاوب على هاي الأسئلة البسيطة. Thank you.